But of all the things that I see when I'm practicing, plaque is one of the most disgusting things. I can deal with the blood, and I can deal with even the infections and the pus, but the plaque is just one of those things that's not very pleasant. So these are some of the things that I want you to remember from today's lecture. So we're going to be talking about how the microflora in the oral cavity relates to dental caries. So you're going to want to know what are the common residents, how the plaque develops, how it affects the saliva. You're going to be learning things about the acquired pellicle, who the bacteria are in the early initial forms of plaque and in the later forms. You need to know how it develops, how it's more than just a thin layer on the teeth, but it's actually a three-dimensional structure. And then how they can vary site by site within the mouth. And then you'll want to know how the plaque relates to the caries sequence. Yes?
Is it okay if we review?
Yeah, we can review it through the lecture or right now if you'd like.
Right now.
Right now? Okay. Instant gratification. All right? So what is the critical pH for the mouth or for enamel? Right, it depends. So it varies inversely to the concentration of calcium and phosphate in the saliva. So when you're talking about it in general, it's around 5.5, but it varies for each individual. Okay. So can the microflora in the mouth create a healthy environment? Yes. Can it create an unhealthy environment? Yes. So they're both true. What is an amphibiotic biofilm?
It goes from not so pathogenic and then it becomes pathogenic?
Yeah, so it depends. It depends on the environment. An amphibiotic biofilm can be either healthy or unhealthy. Let's see, that was 3. What is question number 4? How does fluoride protect the teeth?
Binds to that.
Was that a tough one?
Yeah.
Uh-oh. Okay, so fluoride protects the tooth by combining with the tooth to make fluorapatite.
Wow.
Actually, there's other ways, but none of them are listed. But that's one of the ways that it protects the teeth. When does the microflora, the oral cavity, start to develop?
After birth.
Right, just as soon as the baby's born, it starts to develop.
Yeah.
We'll talk more about that and why that's important. Okay, then we're going to switch from biofilms to the chemistry of caries. So we're going to talk more about pH. So you want to know how that acid-base equilibrium relates to enamel, how changes in pH affect the demineralization or remineralization of the tooth. Critical pH is not constant. It varies. It's different for enamel. It's different for dentin. It's different between individuals. You need to know what makes oral pH drop, and on the other side of that, you should probably know what makes it come back up also. And you want to understand how enamel matures, becomes more resistant to acid. So what conditions are going to make the person more susceptible to decay? What conditions are going to make them more susceptible to calculus? Then you're going to go through the process of how enamel lesions form or caries lesions form. All right, so you guys should have gone to school 30 years ago, because this was our understanding. This is what they told us when we were in dental school, that to get dental decay, all you need is some plaque and some sugar and some teeth, and then you can have decay. And if you wanted to protect the teeth, and you just took away the plaque or the sugar, and then you didn't get any decay. But since we didn't understand it so well, what usually happened was we took away the plaque and the sugar, and we still got decay, so then we took away the tooth, and then we didn't get any decay. But it's changed a little bit since then. So the human body is estimated to be composed of 10 to the 14 cells. So that's a really huge number. I don't know how big it is, but that's a lot of cells. The surprising thing, though, is that only 10% of the cells in the body are actually yours. The rest of them are visitors. So we're going to talk about some of the visitors today. The majority of these cells are microorganisms, and they're found on all the exposed surfaces of the body. And each of those exposed surfaces make up a specific microflora. So when they're talking about an exposed surface, I mean, obviously your skin is an exposed surface, but your mouth is an exposed surface, your GI tract is an exposed surface. It's just a little more hidden than some of the others, but those are all exposed surfaces. So you have microfloras of the skin, of the mouth, the GI tract, reproductive tract, and even within those areas you have specific microfloras within those areas. So when you're talking about the mouth, you have specific microfloras in the fissures of the teeth and on the smooth surfaces of the teeth, and they're going to be different from each other. So they're different, but they share. So they'll transfer organisms or food sources between these different sources also. The reason they're different is that the different areas have different physical properties, different microenvironments, and so it makes it easier for some bacteria to grow and prosper, while it's harder for others. So the environment, you want to remember, the environment is what dictates who's going to be making up that specific microflora. Okay, so the so we're going to be, while there's all of these different microbiomes, we're going to be talking about the oral microbiome, and then we're going to be talking about it in a healthy state, so in a symbiotic state, or so we could describe it as symbiosis, and then in a disease state or a dysbiotic state. Because you never get rid of all of the plaque. I mean, we tell our patients to brush their teeth, get rid of the plaque on their teeth. We measure the plaque scores, but as soon as they set down that toothbrush, that plaque starts growing back again. So you can't get rid of it, and it's probably a good thing, because the plaque, a healthy plaque, does a lot of things that help protect the body. So it promotes the normal development of the immune system, and it keeps unhealthy organisms from colonizing the microbiome. So like you guys read in your reading, that oral microbiome starts forming just as soon as a baby is born. And that first 3-year period is super critical, because that's when the body starts to recognize self. So if the baby develops an oral microbiome that's unhealthy, so if they get they get dental caries transferred from their primary caregiver, their mom or their grandma or their dad or siblings or whatever. If they get a pathological oral microbiome, then they recognize that as normal. And it's harder when they come to see you when they're 19 years old and want to lower their caries risk. It's really hard to change that oral microbiome because the body recognizes it as self. So when you change it, the body's thinking, well, that's not healthy, that's not normal, and it wants to change it back to the unhealthy oral microbiome. So when you have a pregnant patient in your chair and you're treating that patient's oral microbiome, you're also treating the baby's oral microbiome, and you're setting them up to be to have a lower caries risk. So you guys I'll give you another reading later on, but they were able to show that by decreasing the amount of pathogenic bacteria in the mother, they were able to decrease the patient's caries risk as well, or the baby's caries risk as well. All right, so it helps with the immune system. It excludes other pathogenic bacteria from growing just by competing for nutrients or attachment sites. The healthy microbiome can also produce inhibitory molecules to keep the unhealthy bacteria out. So plaque, as much as we say it's a bad thing, it's only a bad thing when it's in its pathological state. In a healthy state, the plaque is actually very helpful to the body. Okay, so in this microbiome, there's more than just bacteria, but bacteria are the most common organism that's populating the oral microbiome. There are more than 700 different types of microbes that can be isolated from the mouth. Not everybody has all 700 of them, but they keep finding more of them. These microbes get their nutrients mostly from obviously from what you eat. They eat the same things. So amino acids, proteins, glycoproteins, all that are found in the saliva or the gingival crevicular fluid. That's where they get their nutrients from. Okay, we're going to be talking lots about saliva, but saliva plays an important role in maintaining the oral pH of the microbiome. And at about 6.8, which is about a neutral pH, this is the optimal level for the growth of microorganisms that's associated with health. You get very much below 6.8, and then you're going to be optimizing the environment for the pathological microorganisms. Okay. Diet plays a role in the development of the oral microbiome, but maybe not as important of a role as what we might have thought a few years ago. Well, it generally has a negative effect on the health of the oral microbiome. There are some things that you can add to the diet that will promote a healthy microbiome. But usually when you eat something, your pH drops, and that is what facilitates the pathological bacteria. Okay, like I said before, the microbiome varies from site to site, even within your mouth. So the tongue, the buccal mucosa, the teeth, they all have different compositions of microbes on them. Even on the teeth, the composition is going to change between the pits and fissures in the area right next to the gingival junction. Interproximal areas will have a different composition as well. But what I want you to remember, because it kind of goes against what we've always thought, but biomes are normal and they're healthy, and we actually need them on our teeth. It's just the pathological ones that cause all the problems. Okay, the most diverse area within the mouth, it occurs on the teeth as far as the populations of the microorganisms. So the dental plaque. If you were to sample your mouth right now, you'd probably get between 12 and 24 different kinds of microbes out of your plaque. So while there's maybe 700 different species of microorganisms found in the oral environment, most people have somewhere between 12 and 27. The interesting thing, or the thing that makes it so that we can treat the dental caries, is that they develop in a very specific pattern. So just within seconds, so before you get your prophy cup off of your contriangle, the oral microbiome starts developing again. So just as soon as you finish cleaning your teeth, the tooth becomes coated with a conditioning surface from the saliva. It has all sorts of different stuff in it. It's called the acquired pellicle, but it has little bits of dead bacteria. It has proteins in it, and it causes a little coating to cover the tooth. This initial coating makes it. This is what the initial bacteria will attach to, and it's mostly there's still Streptococcus species, but they're the Streptococcus myitis and the Streptococcus oralis, so not the Streptococcus mutans. These are the initial colonizers on the plaque, and these initial colonizers are bonded to the tooth by just really weak physical chemical forces. So there's no really strong bonds that are holding this initial plaque to the tooth. Once these early bacterial colonizers get attached, then they set up receptors within their acquired pellicle, and they make it so that the next bacteria that bond to the tooth they have stronger bonds, and it becomes more of a irreversible bond. But that initial bond is just a weak bond. Okay, so once these initial bacteria start to multiply, they start to eat things, metabolize things, and they change the local environment, making it slightly more anaerobic. And as it develops further, the secondary colonizers will bind to the attached bacteria. And this second process is called coadhesion or coaggregation. And so you get more bacteria, more different kinds of bacteria attaching to the plaque. This is called microbial succession. And the second group of bacteria that attach, they attach more firmly, so they don't have that weak physiochemical bond. And as these secondary bacteria attach, they start producing extracellular polymers, and like the glucans and these make the plaque more sticky, make it harder to remove. So this is when you start getting that thick, lumpy plaque that you see on your patient's teeth. But this matrix, made up of the extracellular polymers, the glucans, this matrix can bind and retain molecules, so enzymes, food molecules, charged molecules. So it starts setting up its own little micro environment within the plaque. So these older, more mature plaque are spatially and functionally organized. So they will have so they'll have pantries, and they'll have kitchens, and they'll have storage areas, and it allows some areas to have specific genes expressed in them. It allows for the species to interact, to share information, to share food sources, to share protective sources. So it just makes it harder to disrupt the plaque. So some of the interactions that they can have, like I said, they can share food sources, metabolic components. They can talk to each other, so they'll signal cell-to-cell signaling. They can transfer genes. You especially see this when if you're with antibiotic resistance. They can produce inhibitory molecules to help block out the bacteria that they don't want in that matrix. Okay, so here's kind of a pictorial idea of how the plaque develops. So on the bottom you have the tooth surface, and then just on top of the tooth surface you have the salivary pellicle, that initial covering that happens just as soon as the patients finish brushing their teeth. That salivary pellicle has different proteins in it, has bacterial fragments in it, some mucins, some amylase, so different glycoproteins, different just a layer that allows the initial colonizers to attach to the tooth. So here, where it's circled, you have the initial colonizers. So your strep oralis and sanguinis, strep mitis, strep gordoni. These are the initial colonizers of the plaque. These are your healthy bacteria. Okay? And they make up that first weak bond of the bacteria to the salivary pellicle, to the tooth. Once you get the early colonizers attached onto there, then later on you'll get some of the other bacteria that are attaching to the tooth. So any of those 700 some species, so you're not going to have to remember all 700, or even I want you to understand I want you to know the early colonizers, and then I'll emphasize the bacteria that are the unhealthy ones that I want you to know as well. But we're not going to go through all 700 of them. So here's more of a 3D diagram of how the plaque might develop. So at the top you have the salivary pellicle, and then that's attaching to the tooth, and then you get the initial colonization with the strep oralis, strep gordoni, strep mitis. Once you get that initial colonization, if that's allowed to develop further, then it's going to get some of the other bacteria that are going to start producing the glucans, the extracellular polymers. And you see how it becomes more three-dimensional. And then as it matures, like on the bottom there, you can see how it has channels where fluid can go through. It can wall off certain areas to make them more aerobic and more acidic to allow for certain bacteria to thrive. So our goal is to keep it at that initial colonization stage, not allow the plaque to develop past that into the unhealthy form. But the mature plaque is going to have specific bacterial colonies that set up their own little environment within it. They're going to have fluid channels for nutrients and can go through where they can share genes with other areas of the plaque. It's going to have an extracellular slime layer that kind of protects it, makes it hard for anything within the saliva to get in that it doesn't want to get in. So it creates a three-dimensional environment on the tooth. And once this is developed, it can have its own little environment that's just totally opposite of or different than what the environment that is in the saliva surrounding it. So they'll have anaerobic centers within the plaque. They'll have aerobic centers where they bring in the nutrients. So it becomes very organized and very complex. So here's a different view of it, but you can see that it goes from attachment of single cells to a three-dimensional structure. And once that three-dimensional structure has developed, pieces of that can detach and seed other areas within the oral environment as well. So it can set up colonies in other places. Okay, so like we said before, your dental plaque varies even on the tooth. So in the fissures of the teeth, you're not going to get as much oral microflora developing. The microbiomes that do develop there are going to favor metabolism from sugar breakdown. And it's going to be mostly streptococci with a few gram-negative and some anaerobic organisms mixed into it. Along the gingival crevice, you're going to have a very much more diverse oral microbiome developing. And here as opposed to the fissures, you're going to have many gram-negative anaerobic species and many proteolytic species. The interproximal surfaces, so between the teeth, are a little bit of a mix between those first two environments. So it'll be kind of a little bits of both in there. Okay, so when you're talking about dental plaque, and you're talking about the organisms that are populating the dental plaque, you're going to be talking about some specific traits of these microorganisms. One of the traits that we're going to be talking about is how cariogenic are these microorganisms. So, and it's a pretty simple definition. Cariogenic is just how easily do these microorganisms make dental caries. Some traits, bacterial traits that are related to cariogenicity would be acidogenicity. And the acidogenicity is just how much acid, how acid does that specific bacteria produce with their metabolism. Another trait associated with cariogenic bacteria is acidouricity. So how acidouric is the bacteria? Acidouric bacteria are just bacteria that are able to survive in an acidic environment. So they don't necessarily create acid with their metabolism, but they are able to survive in an acidic environment. Bacteria that are able to produce the extracellular polysaccharides like the glucans and mutans from sucrose. These are very cariogenic bacteria. They make these polysaccharides when they have lots of metabolites available to them. So they can use them to create the three-dimensional structure of the plaque. They can also use them as a storage source. And if the metabolites that they normally have are decreased, then they can use the glucans and the mutans as a storage, as an energy source as well. Okay, so within the ecological plaque hypothesis that we talked about last week, we said that when the environment changes from a normal, healthy environment to an unhealthy environment, then you get an ecological shift in the bacteria, and that ecological shift in the bacteria produces a disease state. So this diagram shows that how you add stress to like excess sugar to the environment, it's going to increase acid production, which is going to change the environment from a neutral pH to a low pH. This is going to shift the microbiome from the healthy bacteria, like the sanguinus and the gordonae, to the unhealthy bacteria, like the actinomyces and the lactobacillus and the strep mutans. And that shift in the ecology of the biome is what shifts the patient from a healthy state to a disease state or a cariogenic state. So it's kind of a waterfall effect. You add a stress to the environment, change the environment, it shifts the oral microbiome and causes disease. So the Caries sequence is kind of evolved from those first three circles that I showed you at the beginning of the lecture to more of a circle. So it's kind of a chicken and an egg circle. There's no beginning or no end point to caries. You can kind of start the caries process at any point in here. So whether you get an influx of cariogenic bacteria, which causes the low pH, which causes growth of more acidic bacteria, which decreases the protective bacteria. Wherever that starts, it can start at any point. So it doesn't always start with just eating sugars. You can start this disease vicious circle at any point. So now that you have a thorough understanding of the oral microbiome, how does this help in how we combat caries, the disease? So the way we understand the caries, the disease, is affects how we're going to treat the patient. So if So back when I was in school, we kind of thought all bacteria were bad, and we just said, okay, you have to get all the bacteria removed from the teeth. And so we tried to brush and floss away all the bacteria. We tried to use antibacterial mouth rinses to kill all the bacteria. And it was maybe not as effective as we wanted. But now that we understand that there's specific bacteria that are affected by the environment, we can target our treatment modalities against those specific bacteria. We can target the environment to try to keep the environment in a healthy state. So if you remember from those series that we talked about last week, any acidouric bacteria can cause the environment to shift. So you don't have to have the two main bad guys, the strep mutans or the lactobacillus, to have caries, because any acidic or acidouric bacteria like the actinomyces, they can thrive in that acidic environment and cause it to shift to a place where the pathogenic bacteria like the strep mutans and the lactobacillus can thrive. So while lactobacillus and strep mutans are two of the best at causing caries, there are hundreds of others that can shift the environment to a place where the lactobacillus and the strep mutans can thrive. So different strategies. So we can try to inhibit the acid production of the plaque. We can try to keep the oral environment at a neutral pH by how often we eat and what we eat. We can target foods that are non-cariogenic. We can try to stimulate the saliva after meals to help keep the environment at a neutral pH. Just about anything that we can do to maintain that healthy neutral pH environment is going to help to prevent the caries. And that goes along with the ecological plaque hypothesis. So maintain the environment, make it hard for the pathogenic bacteria to develop. I took down my clock at the end of the hall. All right, so inhibiting plaque acid formation. So there's a few things that we have that can help inhibit the production of acid. Fluoride doesn't inhibit the production of acid so much as it helps protect against that acid. So it increases the remineralization, increases the resistance to acid. It does have a small antibacterial effect, but not huge. But it's the main role of fluoride is in the remineralization of the teeth. Xylitol, you guys have all heard of xylitol, I imagine at this point. But xylitol is a sugar alcohol, a sugar substitute. The thing that we like about xylitol in regards to caries is it can't be metabolized to acid, and it forces the acidogenic bacteria, the acidouric bacteria, to use the extracellular polysaccharides like the glucans and the mutans that organize the plaque. It forces them to eat the plaque to make energy for themselves. So it helps to break down the complexity of the plaque. It competes for the same receptor sites that the sugar does when the strep mutans is trying to metabolize the sugar. So it kind of clogs up the metabolic pathway that way. And then silver. Silver is bactericidal. We'll talk more about it, but silver is also used to combat plaque formation. Okay, so when we're talking about diet, so there's two things you want to remember when you're talking to your patients about diet. What they're eating and how often are they eating it. So increased frequency of snacking will shift the biofilm from a symbiotic state to a dysbiotic state. So decreasing the frequency of snacking causes a reduced amount of time where the environment is at a low pH. Some specific foods that will not shift the pH in the local environment to such a acidic condition would be like some nuts or string cheese. Mostly, we'll talk more about it later, but any food that has the amino acid arginine in it is going to be broken down into ammonia instead of to acid. So it's going to cause the oral environment to be more basic. And just using rough coarse foods like carrots or anything other than gummy bears, things that aren't sticky to the teeth. If it kind of brushes the teeth for you while you're eating it, then it's less cariogenic. If it tends to if it tends to stick to the teeth, then it's more cariogenic, and it makes sense. But yeah, protein-rich foods, especially that have the amino acid arginine in them, nuts, cheese. Everybody snacks on chickpeas and soybeans, right? But turkey, these all have lots of arginine in them. And some of them are easier to snack on than others. And then what are some strategies for increasing the saliva flow after meals would be there also. But what was your question?
Sorry, what did you say they break down to instead of acid?
Ammonia.
Thank you.
Yeah. So the arginine breaks, instead of breaking down into acid, it breaks down into ammonia. And anybody who's worked with ammonia knows how basic ammonia is. Okay, but yeah, some strategies for increasing salivary flow after meals would be helpful because the saliva brings in components from the immune system to help with the host defenses. It increases the buffering capacity around the plaque, removes some of the fermentable metabolites that are in the plaque. It brings in minerals and other things that will help with remineralization. And increasing the salivary flow will help to return the plaque to a more normal pH level more quickly than lower salivary flow will. So stimulating salivary flow after meals is a good idea. Then whatever else you can figure out to reduce the levels of a plaque and maintain the oral environment at a neutral condition is going to help. Okay, so here's a little summary of what we were talking about so far. But your pellicle formation, that's the thin bacteria-free layer that forms on the tooth within just as soon as you finish cleaning the teeth. And then within hours of that, you get those initial colonizers attaching to the teeth. And remember, this is the the weak physiochemical bond of these initial colonizers. Then these initial colonizers will incorporate a few other bacteria, and this will be your young supragingival plaque. Okay? And at this point, your plaque is healthy. So maintaining the dental plaque at this point is it's a symbiotic state. Once you get a little older plaque, there's an increase in the percentage of gram-negative anaerobic bacteria, and this is when the plaque shifts to an unhealthy state. And it starts to have that thick three-dimensional structure with it's organized and resistant to the saliva, to the oral environment around it. Okay? And then that supragingival plaque is going to seed other areas, like the subgingival areas and those. And this is the disease state of the plaque. So keeping it at that third level there, that young plaque level, that would be the healthy state. And that would be our goal in caries management. So let's see. Let's take 10 minutes, stretch your legs, come back, and then we're going to shift gears and talk a little bit about the kind of the chemistry of the caries instead of just the microbiology.
It's over there.
I can see what you're watching, but I just do like a figure. I'm like, wow.
Hey,
We're just going to.
it's okay.
That's where I dropped the place.
What happened?
Try suggesting new things.
Yeah, she does go over the
And then I stopped and I was like shaking. And I was able to like record it.
Dude, you were still in bed. You were still.
I was still in bed.
Dude, why were you still in bed? I would have.
Oh wow.
Yeah.
Oh yeah, I was chilling in my bed.
I didn't
And I
write
was
anything.
thinking,
Me too. Like, yeah, I was like,
She's like, oh,
dude, I was.
well, yeah, it was for a while. Like, it was shooting very
Yeah,
fast.
dude, the video was like.
Like 30, like a little over 30 seconds. Well, I think I have I felt the
I don't know. That's like a song that
It's like it feels a little too really.
It's hard to get to the heart.
Every night.
Also, song was like,
Other than that, it's just like I'm. Dude, it was so
there's really brown.
And birthday.
I mean, you get valuable information, but I feel like the first hour, or, it's a 2 hour class. I feel like the 1 hour of it is like good information, and then the second hour is just kind of like, you don't have like and so that's kind of but if we're going to be up covers on that it's a practice. Yeah, that's actually
Well, she also has it's a girl. Like in the box, we were talking. I don't know. It's talking against. I don't think How's it and then like sorry, this is the one I see. Okay, yeah, I'll get it. Okay. Sorry. No, you're good. Yeah, I know. Like, I see that for one, but stick around. I'd rather not. Probably asked like
All right, let's get started with the second half of the lecture. So the first half of the lecture, we talked mostly about the bacteria and the biofilm, but I kept mentioning things like pH and stuff because they really go together. So the second half of the lecture, we're going to be talking more about pH, but we're still probably going to talk a little bit about bacteria also because it's hard to separate those two when it comes to dental caries. So in the normal, healthy individual, the teeth are always demineralizing and remineralizing. So that cycle of demineralization and remineralization is normal, and it's part of a healthy tooth. So just like in the past, we used to say that plaque was bad. We used to say that demineralization was bad, but that's not entirely true. Some plaque is helpful, and some demineralization is helpful. It's only when the demineralization outweighs the remineralization. So when that cycle gets out of balance, that's when it becomes pathogenic. Okay. But just remember that in a healthy environment, you are always getting demineralization and remineralization. So it's just part of a healthy process. And just with healthy individuals, whenever you're eating, your pH in your mouth is going to drop. So when you eat, the pH in your mouth drops. But in a healthy individual, that pH comes right back up to where it's supposed to be at a neutral pH. So if your patient is snacking throughout the day or sipping coffee all morning long, that pH just never gets a chance to come back up to a neutral pH. So that's when your when your cycle of demineralization and remineralization gets out of whack. All right, so back to college. And what we're talking about is just basic chemistry. So we're talking about how a weak acid dissociates into the acid and into the ion in an aqueous solution. So you go from acid and the water separate to the acid by itself with the ion floating around in the water. And that's on a really simple level what's happening to the saliva when the saliva goes from a neutral state to an acidic state. But the equation is always in equilibrium. So it's just where those ions are moving, whether they're associated with the acid or whether they're just by themselves. But it's not just, none of these things are destroyed. They just get the form is changed is all. So when you're talking about a tooth, you're talking about calcium and phosphate and hydroxyl ions, and how these are either all bound together in the hydroxyapatite, or when they're in the demineralized state, they are broken up into the individual component parts. So I don't know, how many of you guys played with chemistry sets when you were in grade school? Ready? Yeah, okay, here we go. So an easy experiment is the seeding experiment, where you, or the saturation experiment, where you take some seed crystals, put them into solution, and at a certain point when the solution becomes saturated, then it goes, all of a sudden it goes from a liquid to a crystal. So you can have an aqueous solution. You just add just one more crystal or just a little bit more crystals, and all of a sudden it'll change to a solid state. And so that's kind of what's happening to the saliva, maybe not so dramatically, but that's what's happening to the saliva when we're talking about demineralization and remineralization. So in the aqueous solution, so in your saliva, the hydroxyapatite exists in equilibrium. But when the pH decreases, the equation shifts to the demineralization side, and that hydroxyapatite breaks up into its component parts. When the pH increases, it starts remineralizing, or putting those component parts back together, and so you get remineralization. So at a low pH, you get demineralization, so the calcium and phosphate are coming out of the tooth. At increased pH, so the neutral pH, you get increased remineralization. So the calcium and the phosphate are going back into the tooth. Okay. So when we're talking about the demineralization of the tooth, oftentimes we'll talk about the critical pH of the tooth. So if you're talking about enamel, that's somewhere around 5.5. From your reading, I think everybody got that one, that it depends on the individual. It depends on how much calcium and phosphate they normally have in their saliva, so the buffering capacity of their saliva. So it's not constant. We get stuck on the critical pH of enamel, but maybe more important would be the critical pH of the dentin or the cementum, because especially with patients with periodontal disease, when that cementum on the roots are exposed, you'll get root caries there. Problem is the critical pH of the cementum is is much higher than the critical pH of enamel. Enamel is a lot harder. So the critical pH of the cementum is going to be somewhere around 6.5. So you don't have to be very far off from the normal of 6.8 to already be at a place where the roots are going to start demineralizing. So that's why root caries is such a tough thing to fight. But as the pH drops below the critical pH, whatever it is for that patient or that substance, you get demineralization. If you can keep your pH above that critical pH, then you're going to get remineralization. So in a healthy individual, the saliva and the biofilm are normally supersaturated with respect to the tooth enamel, because the pH in normal, healthy individuals, the pH is much higher than the critical pH of the tooth. So that's why for healthy people, their teeth just don't melt away in their head. But if you get an unhealthy individual with very acidic saliva, you can't do fillings fast enough to keep ahead of the decay. So that's why it's so important that we keep the environment of the mouth in a neutral pH. So here again this kind of illustrates the different critical pHs of some of the structures of the tooth. So again, enamel somewhere around 5.5. Dentin or cementum is very close to a neutral pH, so around 6.5. Reason fluoride works so well is because when fluoride gets incorporated into the enamel, the critical pH drops again. So then it's about 4.5. So it has to be really acidic for people who have teeth that are made up of fluorapatite. All right, so just about everything we eat causes the pH to drop, at least everything that tastes good. But yeah, fermentable carbohydrates will cause the pH to drop. Lots of times people will talk about the Stefan curve. So the Stefan curve is measured plaque pH. So Stefan, he put some little pH sensors on the bottom of a bridge. I don't know how many. Anyway, he got a bunch of people with bridges, put a little pH sensor on the bottom of the bridge where the plaque would accumulate. Then he would make them drink a sucrose solution, and he would measure the pH from those little sensors. So what he measured was plaque pH. So it's probably a little more accurate than measuring salivary pH, because or maybe not as accurate, depending on your point of view, I guess. But from what we learned in the first half of the lecture, the plaque will tend to keep out the plaque tends to be more acidic, so it tends to exclude the stuff that is in the saliva. But it has a similar result. Anyway, so he measured plaque pH, and so he would give the patients a glucose rinse, and within 10 minutes of rinsing with the glucose solution, their pH would go from a normal neutral pH down to a very acidic pH. So the pH would drop very rapidly. The problem was the pH would recover, but it would only recover slowly. So they would spend a good almost 30 minutes with their pH below the critical pH level before it came back up again. So while the pH will drop rapidly, it usually recovers slowly. That is the Stefan curve. Clicker is not clicking. Okay, so some of the things we eat are more acidic, more, they're going to change their environment more quickly than other things. Hopefully nobody is, actually I had, so when I was living in Africa, we would go to some of the local villages and try to help out there. But some of the local treatments for dental caries would be to drip battery acid onto the tooth. So some people do put battery acid in their mouth. Fix the decay in the tooth, or maybe, I don't know if it fixed the decay in the tooth, but it did kill the nerve of the tooth, so it stopped hurting. So I guess it was somewhat successful. But not very far away from battery acid is lemon juice. So your patients who bring in their bottle of water that has a little lemon floating around in it, they're probably demineralizing their tooth fairly strongly. So, and then some patients will suck on lemons, and so that will demineralize the teeth really rapidly as well. Saliva in cow's milk is around 6.8, so that's pretty neutral. Pure water is about 7. A lot of the bottled water that you buy tends to be acidic. So just because it's water, especially if it's coming from the tap, it's probably a neutral pH. But a lot of the bottled water that you get, it tends to be acidic. So if your patients are drinking bottled water and they have a problem with acidic pH, you might want to have them check the pH of their water and just to see. Baking soda obviously is very basic. It seems that when my mom washed my mouth out with soap, she was doing more than fixing my vocabulary, but she was actually helping with my caries as well. Who knew? Okay, some of the other things that causes pH to drop. Xerostomia. So xerostomia or hyposalivation is really tough in regards to dental caries. It takes away everything that protects the teeth. So, and it makes the mouth very acidic. So a patient with xerostomia has a tongue that doesn't look too much different than a dried lake bed. But these patients, they know it. Their tongue is sticking to their mouth, to the sides of their mouth. It's hard to talk. It's painful, burns, it stings. Not good. Rabbits seem to suffer from acid reflux as well. But normally, your patients with acid reflux, you're getting that stomach acid that's a pH of 1 or 2 and just bathing the tooth. So that's why acid reflux is so destructive to the teeth. Patients that purge, that's why they have such problems with destruction of the enamel. So with acid reflux, you'll get little amalgam islands. So if they still have amalgam fillings, if they still have silver fillings, those silver fillings don't erode. So you get little amalgam islands sticking up out of the tooth. So it's just interesting. It doesn't have anything, but the tooth will erode around them. Okay. So when the tooth first erupts into the oral environment, the enamel is very, how do you want to say it? The enamel is very pure, so it doesn't have very many impurities in it. It's just the hydroxyapatite, and that's it. As soon as it erupts into the oral environment, it's colonized by the by the bacteria in the oral environment, and that causes the composition of the enamel to change. So it will, as soon as that tooth erupts into the oral environment, it starts the cycle of remineralization and demineralization. So it gets impurities incorporated into the tooth, impurities like fluoride, for example. And so that will, you'll get fluorapatite forming within the tooth. And this is super important because as it goes through this remineralization and demineralization cycle, it becomes more resistant to decay. So the brand new tooth that erupts into the oral environment is very susceptible to decay. And probably those little fissures aren't fully formed yet. But once it starts going through the remineralization, demineralization cycle, those fissures will coalesce, form together, and the tooth becomes harder and more resistant to decay. So a brand new tooth is more porous, and as it goes through these cycles, it becomes less porous and more resistant. You'd think that a pure, healthy tooth would be more resistant to decay, but actually one that has some other things incorporated into it, like the fluoride and other organic materials, is actually harder and more resistant to decay. So this is very helpful because it makes the tooth less susceptible to decay. So as your patients go through periodontal disease, they'll go through the same maturation process on the roots of their teeth. So as the roots of their teeth become exposed to the oral environment, they're going to go through the same remineralization, demineralization cycle. And if they have a healthy environment, those root surfaces will become more resistant to decay. If they're in an unhealthy environment, then that brand new porous root surface is going to demineralize very quickly because it's not as resistant to decay as the roots that have been exposed for a long period of time already. Okay. So in a demineralization state, so when the saliva is acidic, there's a loss of calcium and phosphate and hydroxyl ions. And this is being produced by the acidic components from the fermentable carbohydrates. So to combat this, in an undersaturated environment, it's going to try to pull minerals from other areas. So it's going to pull to balance this out because this has to stay in equilibrium. So it's going to pull minerals from the saliva. It'll actually pull minerals from the bacteria. It'll pull minerals away from the calculus. So if you have an acidic environment, it's going to actually destroy some of the calculus. It will pull calcium from the calcium fluoride formations that are typically built up on the tooth. And if that's not available, it's going to pull calcium and phosphate from the tooth itself, which is the demineralization process. So when the pH is acidic and it needs more calcium and phosphate ions, these are the sources where it's going to pull those calcium and phosphate ions from. So if it stays in an acidic environment, it's going to start creating lesions in the tooth surface like this. But as it pulls away the minerals from the tooth surface, the solution stabilizes and the mineral loss will stop. Okay. So once that mineral loss has stopped, if the pH in the saliva raises again back to a normal healthy pH, then you get a situation where you have supersaturation of the saliva in regards to the calcium and phosphate in the saliva. Then the minerals can go back to where they came from. So they can precipitate back into the tooth. But it doesn't remineralize the tooth fully. So it's going to partially remineralize these demineralized enamel crystals. If you have long periods of supersaturation and it's remineralized all the teeth that can remineralize, it's going to start forming dental calculus as well. So you don't want your patients to stay in a supersaturated state for a super long period of time, because it's going to cause other problems as well. It might be good for their caries, but it's probably going to affect the attachment of the periodontal structures to their teeth. So I actually heard a lecturer say that calculus protected the teeth from caries. I'm not sure that's exactly true. I think that in a supersaturated state, there's plenty of minerals to go around, so that the teeth are being remineralized like they're supposed to. But there's such an excess that you're getting calculus deposits on the teeth as well. So I don't think that calculus really protects the tooth from caries. It's just in a supersaturated state that it will get calculus formation as well. But yeah, some people will say that having calculus on your teeth will, if you go to an acidic state, the minerals will come out of the calculus first and protect the teeth. Which may be true, but then you're going to start losing teeth from other causes other than caries, so I don't know if you can really say that it's protecting the teeth. Anyway, in a supersaturated state, yes, you will remineralize the teeth, and if you keep staying in that state, then you're going to start building up calculus as well. So just like with everything else in life, moderation. You don't want to. You can have too much of a good thing. Okay, caries lesions start when the demineralization of the tooth outweighs the remineralization of the tooth. So if you're having more demineralization than remineralization, then that's when the carious lesion will start in the tooth. Okay. Once that carious lesion develops, the conditions, the environmental conditions here on the inside of this carious lesion, just like there's different bacteria in the fissures of the teeth and different bacteria on the smooth surfaces of the teeth, there's different bacteria inside a carious lesion as well. And again, they tend to be more acidogenic, more anaerobic. These tend to be obviously very cariogenic bacteria. Okay, here's a lesion that is just stuck to the surface of them, or it's just isolated to the enamel of the tooth. But this is where the demineralization process starts. So for an initial enamel lesion, it's going to make the surface of the tooth more porous. It's going to feel a little bit rough if you drag an explorer across the surface of it. And once the surface of the tooth becomes more porous, then it allows for a subsurface lesion to develop. So carious lesions develop in the subsurface because the crystals just below the surface haven't been going through that demineralization, remineralization process, so they aren't as resistant to this demineralization change. So the lesion will develop more quickly in the subsurface area. Fluoride, if the patient has been using fluoride or been exposed to fluoride, tends to have kind of a superficial effect on the tooth. Unless it's incorporated into the tooth during the development of the tooth, putting fluoride on the tooth only strengthens the outside part of the tooth. So that's why, again, why the subsurface lesion will develop more quickly. Once it breaks through that enamel layer, then it gets into the dentin and it progresses even quicker. So if you remineralize these lesions, ideally if you wanted to remineralize a lesion, you would remineralize it from the very deepest part first and then remineralize it in layers coming out. But that's not the way that it happens. When you remineralize a carious lesion, it remineralizes it from the outside in. So it only remineralizes the outer surface of the lesion. So you'll always have a demineralized layer in the depth of that lesion. So if the patient again goes into an acidic state and they get through that remineralized portion of the tooth, then the decay will progress even faster because they already have that subsurface lesion present. So normally, you've probably all seen a patient who has a tooth missing, and you can see a little brown spot on the proximal surface next to where the missing tooth was. And this is just a non-cavitated lesion that has remineralized just under natural conditions. And these remineralized areas on the tooth are healthy, and there's no problem unless they get subjected to an acidic condition again and the teeth demineralize again. So normally these areas are not an area of concern. Once the decay starts demineralizing the dentin, it's similar to the enamel, except the dentin is not as resistant to decay. So the dentin crystals are smaller and more reactive, so they break down easier in this demineralization process. They don't need, if you remember again, the critical pH of dentin, it's much higher than, it occurs at a much more basic pH, so around 6.5, so it doesn't even need to be that far off from normal for caries to continue to progress in the dentin. The caries process stimulates the dentin, however, and it causes mineralization of the dentinal tubules. And this is also known as dental sclerosis or sclerotic dentin. So the dentin will try to protect itself by remineralizing the tubules within the dentin, and it'll make a hard brown layer of dentin. So it will look like decay sometimes. In the presence of demineralization, the dental pulp also reacts and will start forming more dentin around the pulp to try to protect itself. And this dentin that's from the dental pulp reacting is called tertiary dentin. So it will start calcifying or mineralizing the pulp chamber and making the dentin thicker to try to help insulate itself from the carious lesion. Okay, so as the minerals keep getting sucked out of the tooth in the demineralization process, once it loses a certain amount of the mineral content from the tooth, the three-dimensional structure can't stand up to the normal occlusal forces of chewing or brushing or even if their dentist takes an explorer and shoves it into the tooth. But once it loses a certain amount of that mineral content, it becomes weakened, and then the three-dimensional structure collapses, and this is what is called cavitation. Just when this is the hole in the tooth that everybody recognizes as caries. Okay. But this is cavitation. Once cavitation occurs, and we can't remineralize the tooth, we can't make that tooth grow back anymore. And then you get cavitation, and you get a protected environment for the dental biofilms. So like I was saying, you get another unique biofilm growing in that area. And this tends to be a very anaerobic biofilm, with high concentrations of cariogenic bacteria, and it causes the caries process to progress even quicker. Okay, so once you get into the dentin of the tooth, the dentinal lesions will have different levels of demineralization. The areas next to the biofilm, these are going to be highly demineralized, very soft areas of the dentin. And this is what we often describe as infected dentin. Deeper areas will show less demineralization. They'll still be discolored, but these areas are harder. And these would be, this area is called the affected dentin. So once you have a cavitated lesion, you can't remineralize the tooth, so you have to do some sort of surgical intervention. So the idea is to remove as much infected dentin or remove all of the infected dentin, but leave that affected dentin that still is highly mineralized. The affected dentin retains the collagen cross-linkage, and that structure is still, when that structure is still present, then you can still remineralize those areas of the tooth. Okay, so when we're talking about caries management, remineralization of the active non-cavitated lesion is kind of the goal of caries management. So as long as the carious lesion is not cavitated, then we'll always try to remineralize the lesion. Once you've remineralized the lesion, that surface is sealed. If you drag an explorer across it, it's nice and smooth, it's not rough anymore. But once you have a sealed surface, then it will protect that lower subsurface lesion underneath. So the remineralized enamel is more resistant to demineralized enamel. But for remineralization, you have to have bioavailable calcium and phosphate to remineralize these lesions. And this remineralization process happens more quickly in the presence of fluoride. All right. So lots of times when a dentist sees any dentin involvement, then they're automatically starting to think about operative treatment. So once you have a cavitated lesion, then you have to do some sort of operative intervention. But non-cavitated lesions can be arrested and reversed. So it's a little bit of a paradigm shift because instead of filling all cavities, now we're trying to decide which cavities do we have to fill. Because the non-cavitated cavities, they can be remineralized, whereas the cavitated lesions, that we're still stuck with doing fillings on those. But we know how to do fillings really well, so most of the latest research has to deal with figuring out better ways to remineralize these initial lesions. And that's it for today. So I let you out a little bit better than last week. You guys have any questions? Hard to imagine that I did it that well. There's no questions at all. Okay. This morning when I logged on to Canvas, it said that they didn't recognize my log on, that I'm not in the system. So I don't know. I need to look and see what's on the modules for next week. If there's any PDFs attached to the modules next week, that's what the quiz is going to be over. So next week is saliva, right?
There's nothing in there.
But there's not reading material.
Right. No, because next week is saliva.
Yeah.
So no quiz?
So no quiz. So you get a break.
You can't say that. You can't say that.